All right. So we've heard a lot of awesome stuff today, some very complex models and concepts. So now for something completely different, a stupidly simple model. But even more importantly, I'm going to talk to you about trematodes and how they're objectively the best. That's right, you've let a parasitologist on stage. What's the conference come to? If you're already a trematode true believer, you're probably familiar with the adult, the sexual stage up here. These live in vertebrate final hosts, including hundreds of millions of human beings. So of course, I'm going to ignore them today. And I'm going to focus on the other really important part of the digenean life cycle. It's this asexual stage that infects mollusks. And in some trematode species, we see two distinct body types in a mature infection when we open up the host and look at those worms. We see these big progeny-producing reproductive worms, and then we see these little things. So the little ones really like to fight other trematode species that try to come in and co-infect their host. You can see this little one here. It's just sucking the insides out of a competitor. So violent. And if we look at the thousands of worms inside of an infected snail, and we take a size frequency distribution of those worms, we see this pattern in some species, a bimodal distribution. If these little things were just babies, there are a lot more of them than required to replace those big reproductives. There's an excess that's not contributing to reproduction, but it is contributing to defense. So there are three hypotheses that have been proposed in the literature to explain what we're seeing here. The first, these small worms are maybe a permanent sterile caste that's analogous to the worker caste that we see in social insects. Second, we may have something like a temporal caste going on here, meaning that the age of an individual worm is determining its behavior and function inside of its infrapopulation. And third, there's this idea that what we're seeing here that has nothing to do with castes or defense. This just reflects some kind of a demographic process that is unrelated to social organization. So how could we test these three competing hypotheses? Well, unfortunately, I proved over the course of about 3 years that many, maybe most trematodes are not amenable to lab culture or to experimental infection. So we can only ever observe these infections once at dissection. So how do we solve this kind of intractable problem here? Well, let's start with a model of what's happening inside an infected snail in a trematode infrapopulation. This is the simplest possible model. Worms are born as minors. That's analogous to soldiers. And then some of those minors, they grow up to become reproductives. Some worms eventually die. The answer to our question about these three hypotheses lies in this model, specifically in the arrows. So we can recreate the cyclic graph, something you may be very familiar with, a projection matrix model. And we define a projection matrix of all of the vital rates going on inside of a snail. So growths, births, deaths of worms, that's corresponding to those arrows. And we just multiply that by a current population if we wanted to know the state of that population of worms in the future. It's very easy. It's the foundation of demographic analysis. In the case of trematodes and other parasites, we can actually create an infrademographic model using this exact kind of machinery. So Mike this morning talked about local demography. Well, this is super local demography. Within host, it's infrademography. And we can solve this model here not to estimate the future of the parasite infrapopulation, but to calculate its vital rates. So we just use this model to calculate the parameters in the matrix. And if we know those vital rates, then we can test our hypotheses. So To solve this system, what do we need? We need the current state of that infrapopulation of worms. We can get that through direct observation, crack the snail, count the number of worms inside. We need the future state. Trematode infrapopulations grew with their host. So we can get that information by a host size, by worm abundance regression for each of our target species that we're interested in. Combine that with data on host size. So this is in the literature. And then for a full solution, what we need is to estimate one of the rates in that projection matrix. And we can estimate any of them, whatever's easiest. So I did previous work calculating the death rate for 3 species of marine trematodes in the California horn snail. I estimated these rates for 20 individual infections. That's these points in the bars that you're seeing up here. And you'll see that each estimate has associated uncertainty. So we'll come back to that. And we'll just plug these estimates right there into this mu parameter. So these are death rate estimates. And with these 3 pieces of information, we can fully solve our model. And if we expand it out, we see we've got these 3 simple equations. We can solve them algebraically. We just plug in our numerical estimates of these 3 pieces of information, calculate all of the other rates. It's basic algebra. Just want to emphasize again, this is an incredibly simple model, both conceptually and mathematically. So does it capture reality? I gave this model, when I was parameterizing it, nothing but the death rates. I fed it death rates. So how well then does this model predict actual frequencies of babies, new worm births in real infections? Well, here's the model prediction. The point in the bar, that's a prediction in the credible interval for a single infection across those 3 trematode species we're talking about. How do these model predictions compare to reality? Pretty well. So in all cases, model predictions overlapped with real-world observation. This simple model of within-host dynamics is capturing reality. So it's making predictions that are true, and it's based on 1 observation per infection. So now we trust this model, okay? Remember our core motivating question then. Why do we observe bimodality in trematode infections? And to answer this, we need to compare parameter estimates from this matrix across the range of social organization in trematodes. So what I did, I've got typical infrapopulation stage structures for 7 species of trematodes here. Over on the left, we've got trematodes that invest an outsized amount of reproductive effort into defense. So they have up to 5 of the soldiers, which are the red column, for every reproductive worm in blue. And green columns are intermediate worms that are growing up into reproductives. All the way on the right, we've got species that have no social organization. And they've invest nothing, at least numerically, into defense of their infection. So they're unimodal. So I've arranged them in order of a decreasing minor to reproductive ratio. To solve these matrices, I've made the simplifying assumption that death rates across all of these species are in the same order of magnitude. So if we're comparing across these species, let's look at growth rate. So do strongly bimodal trematodes have super fast growth rates? That might explain bimodality. No, there's no real pattern here in growth rates. What about birth rates? Well, here we're getting some signal. Trematodes with higher birth rates have more minors. They have more soldiers. So, okay, that makes sense. But there's a lot of overlap there. That signal is weak, except in the most extreme cases. So what if we look at the amount of time that a small worm remains small? And here we've got something exciting. So all of the species with pronounced bimodality in their infrapopulation have minors that spend a ton of time in that stage. And as that infrapopulation stage structure approaches unimodality, the time spent in that stage decreases. So what does this mean? practically. It means that trematodes form a soldier caste by the mechanism of developmental arrest. Soldiers are developmentally arrested juveniles. They're little child soldiers. And this is how trematodes build their armies. By using a very simple within-host dynamical model, we could uncover this mechanism that was intractable to direct observation and study. So let's return to our hypothesis. Is there any reason to believe that these bimodal size frequency distributions that we're seeing are a spandrel, that trematodes build up this big crowd of morphologically juvenile worms that are disproportionately aggressive towards enemies, but that this is not a product of selection? Is this an accident? And I think we can discount this. So our model shows that something weird is happening. Soldiers are living for years, but they can grow up in just days. So this seems to be the product of selection for defense. But what about these other two hypotheses that we've got up here? Can the model that we have discriminate between these? Not directly, but it does give us a little bit of insight. So if this were a sterile caste, infrapopulation dynamics would look like this. The minor and the soldier stages, they'd no longer be synonymous. A soldier would be born a soldier, possibly die a soldier, whereas minors would be born and quickly transition to reproductives. But we've got no evidence of this. So in all trematodes known to have soldiers, all of those soldiers, all of the minors, they appear identical. So in comparing those two hypotheses, the more complex model, we don't have evidence for it. So it leaves us with that last proposal, the last hypothesis proposed in the literature, that the age of the worm determines its role. Older worms are reproductive, younger worms are soldiers. Okay, but if we look at the matrix solutions again, how long are those little soldiers remaining soldiers? Years? So we could take this at face value. Or we've got another interpretation, that the transition to that reproductive stage may be an inducible event, say by the death of a nearby clone mate. We don't have the data to parse those possibilities. So either way, looks like we've got kind of a clear winner. Bimodality arises due to the presence of a defensive caste by the mechanism of developmental arrest of juveniles. And we take this very simple infrademographic model to reach that conclusion. And we've got new frontiers opened by this model. So how quickly can trematodes respond to an external threat by making more soldiers? Here are some estimates that are coming straight from the model. 1 to 2 months, they can build a better army if it's needed. And then we can also ask things like, what kind of evolutionary constraints are in place? What drives evolution for defense in trematodes? This very simple model structure allows us to analytically answer some of these questions, or at least try to address them. Okay, here are some really cute worms, and I'm happy to take any questions if there's time.
Hey, so you said that there was no difference between or at least we can't detect any difference between the soldiers and minors, was it called?
Yes.
Morphologically, but do we see any variance in behavior as far as like whether they'll attack another species?
So the variance that we see is between the minors and the reproductives. So reproductives, they don't attack at all. The minors do, and they attack fairly frequently, but the rate of attack seems to be within that minor pool. It's different for each species, so it's not always 100%. But we haven't been able to say, plan an experiment where it's this minor attacked, and now we're going to take it out of that attack, and then we're going to put it into another and see if it does it again. We just haven't had the ability to look at that. But morphologically, they're the same, but I see what you're getting at. Maybe there could be this behavioral difference between two.
Oh, thank you.
Yes.
Hi, great talk. I'm over here in the middle.
Thank you.
Thank you. That was really interesting how you integrated pretty simple observations with a simple model to get some cool results.
Yeah.
I'm curious whether the infra-population structure changes with the snail size class and age class, which is of course correlated to their exposure to different parasites, and also whether you see relationships like a competition colonization trade-off that has been shown in some other trematode species.
Yeah. So as far as the infodemographic structure, that stage structure changing over time, we've got no evidence that it does. So across a range of infrapopulation sizes, so a range of snail sizes, for a particular species, they seem to have a somewhat characteristic structure. That there's a lot of variability. So there's a lot of individual variability here, but we don't see a clear structure that as they're getting bigger, there are more soldiers or there are more reproductives in all species. There are some that we do, not any of the ones that I put up here, but there are some where we do see that as they get older and older, they seem to produce more and more soldiers. This model can actually incorporate that. So under the hood, it's kind of assuming stage stability. But then I did, I'm not screen sharing anymore. I did allow that stage stability to, assumption to be broken, and then looked at how it changes things. And it's kind of interesting because if you say allow the miner or the soldier group to increase disproportionately with relation to the other two, then you'll see, of course, the model solutions will be very different. You'll get some increase in the amount of time, the developmental arrest rate, and an increase in the birth rate, but then you'll get a decrease maybe in the transition rate, like the growth rates. So you'll have kind of this dynamic evolution of the rates that are going on within that host. As far as the competition trade-offs, I don't know that we've got enough data to really answer that, because we only know of just a few handfuls of trematode species that are following this pattern. So it's something that I think we'd need to look at a large number of things and to kind of compare their life histories, since these things have such different life histories.
I don't know.
Yeah,
Thank Dan again.
I'm so excited. Sorry.
Trematodes are fantastic.
They are.
I get it. But we want to. Thanks.